Okay. Jenna, can you see that video I just put up? Is that showing up?
Yeah, we can see it.
Okay, can you hear it at all?
I can't hear it, but it has the captions that I can see.
Okay. Can you hear it now?
Not really. It's just in the background. I think it's picking up from either your earpiece or the.
Is that better? Now can you hear it? Can you hear
that's
it
better.
now?
That's a little bit better. Yeah.
I mean, if nobody else is talking, we can hear it.
You can hear it?
Yeah.
Meredith, can you hear it?
Yes, I can.
You can? Okay. All right, so shut up. Okay. So I'll play those and just have the sound turned way up so you can hear it. Okay. All right. Oh, that would help. Where's Divna? I'm behind the mask. Okay. All right, let me get that started. All right. Time to start. Okay, let's watch some videos about how some of these transporters work. First, the sodium potassium pump, and then others. There's a rather detailed and somewhat long video about the voltage gated potassium channel. Also, the person narrating it is obviously reading from a script, and they're a little bit monotone, but the pictures are really good, and I'd like you to try and view it and stay awake. So do the best you can. This one is pretty interesting.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites with 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate bonds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this equal movement causes an electric gradient to develop across the plasma membrane of the cell. And no cellular processes, including the generation of nerve impulses, use this electrical gradient.
Okay, that is sodium potassium pump. Now, this one is about ABC transporters. Now, one of the first ABC transporters that was studied in great detail was the so-called P glycoprotein. Why was this protein so important? Well, turns out that cancer cells overexpress it, and when cancer cells overexpress it, they take your therapeutic agents and they pump them right out of the cell. So high expression of P glycoprotein was linked to very treatment stubborn, to put it in colloquial terms, highly resistant cancer cells. So how to gum this up? Well, people studied it in great detail. That's why there's a lot known about P glycoprotein and knockoff about ABC transporters in general. So let's look now at the P glycoprotein and ABC transporters in general.
Transmembrane drug efflux pumps can remove chemotherapeutic drugs from inside a cancer cell, thereby reducing intracellular drug concentration, which generates resistance to the cytotoxic effects of the drug. Certain drug efflux pumps can extrude multiple types of drug molecules, creating a stable multi-drug resistance, or MDR. P-glycoprotein, PGP, is the most prevalent NDR transporter, and it is expressed at elevated levels in many kinds of cancer cells, especially those that have survived the chemotherapeutic treatment. The space-filling model of P-glycoprotein shows the drug molecule, colored in pink, bound to the pump's drug-binding pocket, colored in silver. Such a drug molecule will hit the pump and its drug-binding pocket from the cytosol through the open portal seen at the bottom. Energy is required to pump drugs out of the cell. P-glycoprotein is an ATP-dependent transmembrane protein. An ATP must bind to the interior nucleotide binding domain in order to pump a drug or toxins out of the cell. Switching to a ribbon representation of the pump, we can observe the dramatic conformational change in the pump caused by ATP binding. Drugs or other toxins enter the pump and attach to the drug binding pocket, which results in an ATP-dependent shift in the conformation of the entire pump. The drug is then extruded into the extracellular space. Rotating the structure 90 degrees in the same plane, we can observe the pumping action and the opening and closing of the cytosolic and extracellular ends of the pump. In a third view, we observe the pump through its channel, looking down from the extracellular side.
Isn't that cool? I think that's really cool, but I'm very strange, as you well know. So, very good. Now here is the video about voltage-gated potassium channels. This is long. I'll cut this probably at about 5 minutes. It's very good, but again, the reader is a tad monotone. So drink your coffee, take a sip, and just stay awake, okay?
Potassium ion channels are the KV channels of the protein involved in the conduction of action potential. Before I get into the important structural features of this protein, I wanted to do a brief review of action potential. At action potential, a relay signal causes a change in the membrane potential. As the membrane potential reaches a threshold, the voltage-gated sodium channels will open. As a result, sodium flows into the cell. This is the beginning of depolarization. Once the membrane potential becomes sufficiently positive, the sodium channels will close and the KV channels will open. This marks the beginning of repolarization. At this point, the KV ions will flow outside the cell and contribute to the resetting of rest membrane potential. Thus, the regulation of the KV channel must be such that it opens at a more positive membrane potential and closes at a negative membrane potential. So how does the KV channel open and close at the appropriate voltage? The KV channel's voltage sensitivity is based on the ability of specific residues to change position in a manner that is coordinated with the change in membrane potential. During action potential, ions move across the membrane, which changes the membrane potential. As a result of flux, the electric field across the membrane changes. From physics, we know that electric fields can do work on a charge. In other words, in the presence of an electric field, a point charge can be translated from high to low potential. We can apply this principle to special key residues on the KV channel, which are located on the S4 helix. In this slide, I basically zoomed in on voltage sensing domain to show the difference in structure between the open and closed conformation. When the electric field changes as a result of the conduction of an action potential, arginine residues 294, 297, 300, and 303 can change orientation and can result in the transition from open to closed conformation or vice versa. Because residues are critical to the KV channel's voltage sensitivity, they are one of the most highly conserved residues in the protein. Although the exact mechanism of voltage sensing is not fully understood, the following is one well-accepted theory. First, positive charges flow into the cell during depolarization. As the intracellular space becomes more positive, the positively charged arginine residues that I mentioned in the S4 helix are thus repelled from the intracellular space. As a result, arginine 294, 297, 300, 303 all tilt away from the intracellular space into the extracellular space. The changing position of these key residues causes an outward displacement of the S4 helix. This induces a conformational change in the entire protein. As a result, the K channel adopts an open conformation and potassium ions can flow outside of the cell. In order to close the K channel, effectively the reverse of what I described occurs. As potassium flows out of the cell and down the gate, the intracellular environment becomes relatively more negative and the extracellular environment becomes relatively more positive. It's important to remember that the sodium channel also closes at some point in the process. At a certain point, the positive arginine residues become more attracted to the more negatively charged intracellular environment than the extracellular environment. As a result, the arginine residues in the S4 helix move 15 angstroms towards the intracellular environment. The change in position of the arginine residues induces the conformational change in the KV channel, which causes the protein to revert back to the closed conformation. According to this proposed model, this entire process of opening and closing is hypothesized to not require the input of additional energy. Here, the conformational changes in this model are driven by favorable electrolytic interactions between either the intracellular or extracellular environment and the key residues that I mentioned. So, what mechanisms are in place for the K+ ion to pass specifically? On the left, we have an image of the KV channel in the conformation. The potassium ions can pass through the pore. And the figure on the right demonstrates exactly where the potassium ions bind in the protein. The potassium ions are represented by little green dots. They're hard to see, so I've added arrows that point to their location. In physiological conditions, the K+ ions are hydrated by water molecules. Water is polar, partially negative oxygen is attracted to the positive potassium ions. But the potassium ion cannot pass through the pore when it is still associated with the water molecules. Thus, in order to get K+ potassium to the pore, the KV channel employs a clever physical mimicry. The residues that line the KV pore actually mimic the ion's interaction with water. K+ binds the oxygens in the hydroxyls of the threonine residues and the oxygens in the carbonyl group of the backbone. These residues are a part of the TVGYG sequence. This sequence is in fact highly conserved among species as it allows for these oxygen atoms to be oriented in a way that they mimic how the potassium ion is solvated. This pore has selectivity for potassium because other ions are unable to form as many favorable interactions when not associated with water. So it is less favorable for these other ions to be solvated in order to pass through the KV channel as a result of fewer stabilizing interactions. Thus, other ions are less likely to have the KV pore. An interesting story about this protein is that Waters et al. Actually observed ovarian species of KV channels in a Filipino family.
Okay, that's where I'll cut that off. Alrighty, and okay, those of you zooming, is it moving?
No.
No. Now is it moving?
No, unfortunately.
No. No, let's try it again. Darn. Okay. Now is it moving?
Now it's moving.
Moving, okay. All right, so the young lady, assuming she was a young lady, based on the sound of her voice, sounded to me like she was young. Maybe she's 2 years old, I don't know. But the S4 has those arginines, and it's a change in configuration of those arginines in response to the change of the membrane potential. As the voltage-gated sodium channel opens, sodium will now pour into the cell. That now causes the membrane potential to become more positive. That's going to cause those potassiums now to move away from the more positive inside the cell. Normally, with the membrane potential, the inside of the cell is relatively negative. With a more positively charged internal cell, these args now will move away. That will cause the channel now to open. Movement there is going to send a conformational change throughout the whole channel, and that is going to set up a movement that will open the channel and now will allow the potassium, which is hydrated, to bind to the portions of the channel that will mimic its interaction with water. Because of those interactions with water and their geometry for the dehydrated potassium ion, the delta G of binding is very low, and the delta G of passage is very low. Smaller ions don't have that beautiful fit. You don't get that tight interaction with the components of the channel. They therefore are going to bounce in if they bounce off and bounce out. There's too much energy required for them to go through the channel. That's why this is specific for potassium and not sodium, even though sodium is smaller.
Does the
Yeah. Oh, yeah. Yeah, and there's, this is meant to give you a kind of bird's view of the difference. It's a 60%, 64 difference in potassium concentration outside the cell relative to inside the cell. That's going to also get through, but the fact that potassium gets through and not other ions is due to this mimicry. That was a good way to put it, a molecular mimicry of that particular sequence with threonines and glycines and so on and so forth will mimic its association with water. Therefore, the pass-off from water to the channel, it kind of fools the ion into thinking it's joining water and going through the channel. And it fits it really quite specifically. That's why this channel is specific for sodium and not for anything else. So here's the channel from the side, and our S4 is here, and we can see the bits of S4 and S5 that change conformation relative to the rest of the protein. That is then going to cause a change in the rest of the protein as a result, essentially, of the movement of those because of those strategically placed arginines. Remember what the video said? Arginines are conserved in potassium channels, be they from rat, be they from mouse, be they from squid. They all have those arginines. Same thing with that filtration channel. That specific sequence you mentioned, that's common. To rat, to mouse, to humans, to squid. All potassium channels, voltage-gated potassium channels, have that particular sequence. So if you want to watch that again, it's on Blackboard. Just hit the link, kick back, drink some coffee. If you don't like coffee, some other beverage, and watch it as many times as you like. Here's a view from the top, and in a cartoon showing essentially these portions that will get tweaked, that will then tweak the rest of the channel as well. And then a view from the inside face, and so on and so forth.
So that, the forward line that is, that's on the S6 domain?
It's on the S6 and S5. Both S5 and S6. It's shared.
Could you repeat what she asked for us?
Irma asked if the filtration sequence, which helices it was associated with. It's associated with S6 and S5, and also some of the loops in between. So it's shared by different parts of the protein. It's not restricted to one specific part. The sodium-gated channel works exactly the same way. In fact, all voltage-gated channels work this way. You have a sensor. That sensor changes conformation, becomes a channel. When you go back to the original memory potential, that sensor goes back to its original configuration, closing the pore.
So the sensor, does it have different residues?
Yeah, it doesn't have, it's not necessarily arginine, but it has a positive, usually a positively charged section. And that's usually the part of it. Notice too, those arginines are very close together. So they're clustered. They're going to constitute a positive charge over a very small part of the protein. That's also very common. Ligand-gated channels are a bird of a different feather. They're going to open in response to being bound by a small molecule. And this binding will take a channel that was closed and now open it. The channel that has been most heavily studied in this case is the nicotinic acetylcholine channel. And the source for this channel has been the electric eel, because it turns out that the electric organ of the electric eel are modified muscle cells. And those modified muscle cells are loaded with these nicotinic acetylcholine receptors. So if you want lots and lots of this channel, you get it from the electric eel. And when I was an undergraduate at UC Davis, a guy named Mark McNamee, his lab worked on this channel, and he had a big aquarium that's full of electric eels. And every first year graduate student who joined their lab, they got the job of picking out the eels. When you stick your hand in there, you can feel the electricity. When you grab the eel, you have to make sure your hand's in your back pocket. If you're holding metal or something, you're really going to get it. So you make sure you stick this hand in your pocket. It's just like, any of you done electrical work? Any of you? Your husband has, I'm sure. Well, electricians are told if you're dealing with circuits, you don't know they're hot, you stick your hand in your back pocket. That way you're not touching something. So you get a shock. You don't go all through your body and you really get it. Yeah. Kick the ladder and then, yeah. Yeah. Usually what they tell you in school is electrical training is that when don't use two hands, stick that hand in your back pocket and use the one hand. You'll just, yeah, and you'll be fine. You have a pacemaker. In which case you shouldn't be an electrician in the first place. They said, okay, hand in the pocket. Then they had to grab the eel. The other thing about eels, you got teeth too. They have to shock you. They bite. They bite hard. So these guys, that was their indoctrination into the McNamee lab. And so this channel has been extensively studied. And the channel comes in lots of different shapes and sizes. But the one in muscle has four different subunits. Has an alpha, a beta, a delta, and a gamma subunit. Two of the alpha subunits. So it's a pentamer, but it's a heteropentamer. In your brain, you have versions of this that are homopenters. They're all made of the same particular subunit. So each of these subunits has 4 transmembrane domains. 4 transmembrane domains. And they form this little battery, I guess you could call it. And each of these are designated M1, M2, M3, and M4. And as you can see, the two ends both stick inside the cell. And the subunits they have a lot of similarities in some ways. Now, the acetylcholine binds in between the alpha and the delta and the alpha and the beta subunit. And these particular subunits all have M2 facing the channel. M2 facing the channel. That is significant. Why is that significant? Well, turns out M2 has a bunch of very bulky leucine residues that stick into the channel. Leucine is hydrophobic. So can cation ions easily go through that channel? No, channel's blocked. So we have our little battery here. Here's M2, leucine essentially sticking out right into the middle of that channel, and that channel is blocked. So each of those each subunit there, alpha, beta, gamma, delta, whatever, they all have those 4 transmembrane domains. They all have M2 facing the pore. And M2 has these bulky leucine residues gumming up the channel. All right, you bind your ligand. Here's what happens. And you bind the ligand, these subunits do the twist. And they're going to twist this way. Now, the bulky leucine residues go this way, and you now have a portion of the helix that is hydrophilic facing the channel. Now you've opened the channel. And as long as acetylcholine stays bound to this channel, it will remain open. When acetylcholine dissociates, it'll snap back. Other ligand-gated channels work in a very similar way. So the GABA channel, which is a chloride channel, also rotates when GABA binds, opening it for chloride. So this turns out to be a motif that was confirmed with other channels. They work about the same way. And remember, many of these channels are targets for drugs. So your GABA channels, for example, have you ever heard of drugs called benzodiazepines? What are they used for? Anxiety. They're used for sleep. They're used for decreasing excited states and things like that. They're used for catatonia and what have you. They're used for lots of things. Bind the GABA channel. What else binds this channel? Do other drugs in this channel? How about muscle relaxants? Muscle relaxants like the kind you give during surgery. You have to give them. Otherwise, you're going to cut into somebody and their body's going to just twitch. You want that happening during surgery? I don't. So you give a muscle relaxant. Those muscle relaxants were designed based on the structure of this channel. So work done in an electric eel led to information that led to the design of drugs now used in surgery, of drugs now used for anxiety disorders, and so on and so forth. So again, basic research led to more applied research. Somebody trying to ask something?
Yeah, I was going to ask, is gabapentin similar to what you were talking about previously?
Yes, the gabapentin binds to the voltage-gated calcium channel.
Okay.
So does pre-GABA. Dane asked if gabapentin also binds to the GABA channel. Based on its name, you expect it would, but it doesn't. Binds the voltage-gated calcium channel. So does pregabalin. The trade name is, don't tell me, Lyrica. Gabapentin is Neurontin. But now it's available as a generic. Yeah.
So what causes the acetylcholine?
Cause a twist? To cause a twist?
No, once it binds,
Uh-huh.
it will keep it open
Yeah.
until it dissociates. But what
Right.
causes it to dissociate?
The fact that it binds and lets go, binds and lets go, binds and lets go. It doesn't form a covalent adduct with the channel. It binds, it has a reasonable affinity, but it doesn't stay bound indeterminately.
So it's the same thing as like enzyme inhibitors. They'll bind for a little bit.
Yeah, you could kind of, sort of, yeah. Oh, well, reversible enzyme inhibitors. Yeah, exactly. So the twisting opens the channel. Sodium comes in, calcium comes in, potassium leaves. And that's actually what that channel is for. And your other channels work on very similar principles. So your voltage-gated channels will open when the membrane potential achieves certain value, and they are responsible for the action potentials you see in neurons. And they, as a rule, are going to have 4 homologous domains. Each of those homologous domains contains 6 transmembrane domains. And this is very common. They're a repeat, essentially, of a motif, usually about 4 times. And in each domain, you have an extracellular loop between 5 and 6 that is very important for selectivity. And each domain also contains an intracellular loop between each of these various domains as well. And helix 4 in every one of these voltage-gated channels seems to be the voltage sensor. And helix 6 is the activation gate. So here's the structure of the sodium channel. And once again, 1, 2, 3, 4, 5. And then here's our activation gate. There's our voltage sensor, helix 4. Here's 5 and 6. There's part of the filtration filter. And then down here, elements of 5 and 6 as well. Here's part of the activation gate. Right here, helix 6. And then the inactivation gate down here, which is connected to helix 6. And once again, just like with sodium channel, 5 and 6 line the channel itself. And they're responsible essentially for the selectivity of the channel. And 4, once again, plays a role in whether or not the channel will open or shut. That's the one that detects changes in the membrane potential. And helix 4 essentially will move up or down depending upon the membrane potential. And the membrane has to get to a value that's known as threshold. When it gets to threshold, the activation gate will open, and that will open the channel. And once again, you have positive charges in that helix 4 that are going to rise and fall and what have you. The inactivation gate, the little ball, is heavily charged. It's going to pull away from the channel pore. When you get the membrane getting the threshold, it's away because the membrane starts you get less negativity inside the cell, becomes a little more positive. That's going to draw the negatively charged inactivation gate away from the channel as well, and sodium flows into the cell. And when it gets up to a certain level, helix 4 slides back down again, but that's not before the inactivation gate has closed the channel. Much of the structure and function of these channels were also elucidated by mutants in various genes that encode these channels. And because of their studies of these mutations that cause a variety of genetic diseases, that's kind of led to better understanding of the actual physiology of these channels. Also, people have worked using patch clamping studies with cells that express the mutant channels. And so the electrophysiology of these channels has become extremely well defined as well in some cases. You can also use various toxins that are known to kill these channels. So, for example, saxitoxin comes from a dinoflagellate, Ganiala. Ganiala is what causes red tide in the ocean. If you pick up a handful of ocean water when it's red, they'll be full of these dinoflagellates called Ganiala. They essentially, when they die, they release this compound called saxitoxin. This unfortunately gets into the fish, especially filter feeding animals like clams, things like that. If you eat it, you tend to get some tingling in your lip, and then your sodium channels, voltage-gated sodium channels are inhibited. And then you lose the ability to breathe, and you die. So don't eat fish or clams or oysters or whatever if they've been around the red tide anywhere. Then there's a very famous puffer fish, tetrodotoxin. That is a potent inhibitor of the voltage-gated sodium channel. And this is a very interesting compound. I showed this once to Dr. Baldwin. I think, let's see, after 20 minutes, he got done salivating. He was thinking of great ways to make it and isolate it and put students on it and what have you. To which I told him, I don't think we should be making something quite so poisonous. And he stopped salivating. And then this compound is concentrated in the liver and the stomach and the gallbladder of the organism. It actually probably needs to be, it's made probably as a precursor that needs to be activated by microbes, possibly microbes in the GI tract. If you eat pufferfish, it has to be prepared by a person who goes through a very rigorous training, very serious training program to be able to prepare the flesh of the pufferfish without the flesh getting contaminated by the liver and the stomach and what have you. The flesh is cut into little paper-thin slices. It's called fugu sashimi, and it's served raw like other types of sashimi. And in Japanese, they apparently have a saying, roughly translated, goes something like this: if you eat fugu, you're a fool. If you don't eat fugu, you're a fool. Because the thrill of eating it, also apparently I've been told, it is extremely tasty. That's been told. Never had it. Anyone here ever had it? Okay, well, all right, and it was good. Yeah, well, okay, yeah. I just have this thing, just can't eat raw fish. I had too many parasitology classes. I just can't bring myself to. Just can't bring myself to do it. Nope, won't go there. So that is pufferfish toxin. Again, very, very powerful inhibitor of the voltage-gated sodium channel. Yes.
But how does that work? Because you have enough. Is it because the potassium channels now start to open and start to get the potassium out?
With the potassium channels, you must remember that the sodium channels close, and so the with the potassium channels, concentrate on what I'm doing here for a second, or I'm going to be in deeble. Since that, so the come on, there it goes. So the memory potential you get the threshold and then you depolarize. So right about here, So you're at resting here. You're somewhere around minus 75 or so millivolts. That's your rest potential. Some stimulus gets you to threshold. And then threshold is about minus 55 if you're a neuron. And then voltage-gated sodium channels open. When you get to about plus, it's on the arms about plus 20. Those sodium channels will inactivate because the more positive charge on the membrane potential will cause those potassium channels to open. When they get up here, essentially, your sodium channels have started to close. Now, potassium flux outdoes sodium influx. And so the channel now, the membrane potential is now going to come back down. So that's essentially, it's now going to restore the membrane to its original charge, and that's going to cause the sensor now to go back to its original conformation. That's what closes it. Because it's now back at its original action, and then goes here. So this is called depolarization. This is called repolarization. And this is repolarization. With the sodium channels, it gets to a certain point where an activation gate, which is negatively charged, now is attracted to that more positive charged membrane, and it jumps up and it jumps to the channel and shuts it. And that is going to prevent egress of sodium. And so then these potassium channels are open, and then eventually, because no more sodium can come into the cell, the potassium is leaving, it now reverses. And then when it gets to just around about threshold, now the sodium channel, the sensors go back down and it shuts for good, and so on and so forth. All right. Are the slides moving?
Yeah, they're moving.
They're moving, good. All right. When we started this class many moons ago, it seems now, we talked about how biological systems do not violate the laws of chemistry and physics that describe chemical reactions and the behavior of molecules in our universe. Life was thought to be an exception to those rules for a very long time. Now it's clear life is not anyway an exception to those rules. Instead, life is able to be maintained because of a constant influx of energy. So biological processes, reactions, events, transformations, whatever, they obey the laws of thermodynamics. They obey the laws of thermodynamics. At first glance, they may appear not to. When you start looking at them in more detail, it's exceedingly clear they don't violate these laws. They're not an exception to these laws. So let's review the laws of thermodynamics. It's been a while since Chem 1. You may not quite remember what they say, so let's review. Law 1, for any physical or chemical change, the amount of energy in the universe remains constant. You can move energy around, but you can't create it. You can't destroy it. Can't create it, can't destroy it, you can just move it around or change it from one form to another. And in reactions, it's usually the formation and the breaking of chemical bonds, or the changing of something from one state to another, or something like that. Second law of thermodynamics: for all natural processes, the total entropy of the universe increases. Can you have local areas where entropy is decreasing? Yes, you can, but that's usually at the expense of entropy someplace else. Entropy decreases on the Earth at the expense of entropy increasing in the sun. That's how we explain the local decrease of entropy, excuse me, on planet Earth. Disorder, right. So for our purposes, there are three quantities we want to keep in mind for thermodynamics. So there's body. The building is obviously named after him. I don't think so. That gives free energy. And then this is calculated using the enthalpic change, delta H, and the entropic change, delta S. The enthalpic change, delta H, entropic change, delta S. So Gibbs free energy, very simply, refers to the amount of work that can be done during some sort of reaction. Might think of a reaction just as A goes to B, but a reaction could also be A is outside the cell and then is moved inside the cell. That could be a reaction too. So try to broaden your perspective of reaction. Transport could be a reaction, for example. So it's the amount of work available to do work during a reaction at a constant temperature and pressure. This is highly simplistic, but we'll complicate things as we need to. So, exergonic reactions. Exergonic reactions have a minus delta G value. They are thermodynamically spontaneous. You don't need to put energy into them, theoretically, for them to go. Endergonic reactions have a positive delta G. Positive gives free energy. You need to put energy into them to get them to go. Enthalpy refers to the change in heat content of a system. Does it cool down, heat up, so on and so forth. Now, biological systems, by and large, don't use heat for an energy source. Heat's a byproduct of reactions. It'll be used to keep organisms at a constant temperature. That creates an environment, a temperature at which reactions will occur. But heat is not something that's used by biological systems. So I know in chemistry, you do a lot of heating things to get things to go. Yeah, to maintain. But it, no, because it just creates an environment. The reactions don't use the heat. It's just the enzymes require that particular optimum in order to work. Remember, the enzymes speed up the reactions. They don't take reactions that don't go and cause them not to go. You still need to put energy into those reactions. That energy is put in the form of ATP. or as we'll see sometimes concentration differences, but not heat. You don't tend to use heat. So exothermic, exothermic. Exothermic means it gives off heat. It gets hotter. Endothermic. It takes in heat, it gets colder. Removes heat. And then entropy just measures disorder. If the system becomes more ordered, entropy is decreasing, then your delta S is negative. Becoming less ordered, entropy increases, delta S is positive. So think about this with me. If you're degrading things, you're making things less ordered, right? Your delta S in that case will be positive. If you're building things from precursors to bigger molecules, your delta S is going to be negative. So we can relate these three quantities with this equation that you've seen before, I'm sure. Might still be kind of wallowing in hate of it. Get over it, because you're seeing it again, where your delta G is equal to your output change minus the temperature in Kelvin, the Kelvin, times delta S, times delta S. Now, let's relate this to our reactions, because reactions, doesn't matter if they're inside cells or outside cells, they still obey the laws of chemical equilibrium. So the progress of the reaction, every reaction, can be described by its equilibrium constant, KQ, and you've seen this before, just run through this. So this is your reaction, then your KEQ is equal to this concentration with that power times this concentration raised to that power divided by this concentration raised to that power times this concentration with that power. At equilibrium, that's your KEQ. And as you can see, this is big. Your reaction tends to go towards products. If this is small, your reaction tends to not go towards products. This is a feature of the reaction that's inherent to the reaction. If you put this in a cell, does being in a cell change this? No. The cell has to work with what it has. And it does. And it does very effectively. So our delta G, that's the energy change, and that is the energy change that's needed in order to push the reaction to equilibrium. That's what the free energy change is for the reaction. And we express that as delta G with this little degree sign, or delta G0. Delta G0 is not very useful for biochemistry. So we're going to use a different standard because, first of all, the reaction has to occur in water. Concentration of water is 55.5 molar. Secondly, has to occur at pH 7. So we're going to have a slightly different number. Yes, you can look this up in a book, but we're going to now re-standardize this, and we're going to get something called delta G with a little prime and then a degree, or delta G/0. This is your standard biochemical free energy change. Standard biochemical free energy change. We used to use a P-Chem textbook here by a guy named Alberti. Alberti was the guy who experimentally discovered a lot of these numbers for various biological reactions. We don't use his textbook anymore. But Alberti, his name kind of features large in the physical biochemical research because his lab did just really, really precise and endless work deriving these numbers. These numbers are well understood. They're not guesses. They're numbers that are known to a couple of significant digits usually. So the relationship between delta G and KEQ is well understood, but because it's the standard biochemical free energy change, you're going to have a KEQ that is significant for biological reactions under biological conditions, pH 7 in water. So it's called K with a prime EQ. K prime EQ, and this is the equation that relates the equilibrium constant with the standard biochemical free energy change. So it's minus universal gas law, gas constant, times temperature, that's in Kelvin, times the natural log of the K EQ with a prime. And your book has some numbers showing you essentially the relationship between these, and they expressed either in kilojoules mole or in kilocalories per mole. Either one will work. If I give you problems, I will usually ask you to use one or the other. If I don't, you can use either one. I will usually ask which one. But if I don't, which I don't always, then pick one and use one. I don't care which you use. Just keep in mind that this number is going to be different if you use kilojoules versus kcals. Keep that in mind. So in your chemistry book, your inside folder of your inside, you open your chemistry book, there's those tables. R is in there, and it usually gives two values for R. One is kilocalories, the other is kilojoules. So make sure you use the right one. That's all I ask. These numbers are well understood. They're not guesses. They've been experimentally derived and known to a great deal of precision. And this is something that's inherent to the reaction. Doesn't depend on the means by which the reaction progresses. It is a function reaction itself. It just depends on the products on the reactants. That's all it depends on. And it is a constant. Now, Friday, because Wednesday we have a test, we'll talk about this number. This is a different number. This number depends on concentration, products, and reactants. All right, test is Wednesday. 50 questions, 49 multiple choice, 1 fill-in. And you're going to have to draw some structures. So make sure you know those structures. Okay? See you Wednesday.
You said we have a quiz on Friday.
Yeah.
What's different than the open? Just so the gates, the channels, the pumps, all that.
No, it's going to be on. 13, just the part we've covered. 13, just the part we've covered. 13, just the part we've covered. Okay, Quiz Friday over chapter 13, just the part we've covered. Okay, and Jenna, are you going to be in Wednesday? No, you can't. So make arrangements with Mrs. Schaefer to have her send you the test. Sorry?
I will be
I
here
can't.
Wednesday.
You will be here Wednesday.
Yeah.
Okay, all right.
Because I'm
Perfect.
not actually in quarantine. I just am waiting for a test result.
You're waiting for test results. Okay, just keep me posted on test
Okay.
results and all that, okay?
Will do.
All right, gang. Ciao.
All right.